Thank you for your coming. I am pleasure to talk a keynote in the first DNAQUA conference. I'm Hideyuki Doi. Today I want to talk about eDNA methods for the analysis of macroorganismal populations. So eDNA can provide us the information of macroorganismal populations. For example, species distribution and abundance biomass. This is a new tool for ecology and biomonitoring. Today, I want to talk about these 5 topics and like a flash talk because I have no much time today. So firstly, I want to introduce the biomass estimation by eDNA, and then talk about sampling and on-site methods, and sedimentary DNA analysis for time machine biology. And I want to introduce some of our meta barcoding studies. And finally, I want to summarize eDNA method standardization. Firstly, almost 10 years ago, we conducted the field experiment using these ponds with introducing the common carp like this. And after the experiment, we measured the eDNA concentration of these common carp and their biomass. This is result. X is eDNA concentration, DNA copies per liter, and Y is the common carp biomass per liter. So this graph showing the carp eDNA was positively correlated with their biomass like this. This indicates the eDNA can estimate the fish biomass. And also we applied this method for the field. We estimated the abundance of sweet fish in this river with comparing between the eDNA concentration and abundance counting by the snorkeling survey. So this is the result. X is abundance of this sweet fish. Y is the DNA concentration. We conducted in 3 seasons, May, July, October, and in all seasons, we found the eDNA concentration positively correlated with this sweet fish abundance, like this. So these 2 studies was conducted using the real-time qPCR, and we also applied the biomass estimation by eDNA using digital droplet ddPCR. It's kind of next generation PCR with absolute quantification using that PCR mixtures divided droplets, and then we measured each droplet using flow cytometry, and we evaluate absolute quantification of DNA concentration. We set a mesocosm experiment using the common carp with different number of individuals per tank. And then we measured this eDNA using qPCR and also ddPCR. This is result. X is eDNA concentration, Y is CB. CB means that coefficient of variance. We found that qPCR have higher CB, especially low eDNA concentration here, but ddPCR have lower CB values than qPCR. That indicates ddPCR outperformed qPCR in quantification for fish biomass with low measurement variance. So secondly, I want to introduce the water sampling and on-site methods for fish distribution. We developed the breachable gears for UAV drone water sampling methods for eDNA surveys. This photo showing our drone sampling gears like this. We automatically collected the water from the lake or stream or marine systems using this drone system. And we compared result with the boat sampling. We found that eDNA detection levels of UAV sampling were similar to boat sampling. So this figure showing the comparing the UAV and the boat sampling, and we found a similar eDNA concentration in these two invasive fish species in the reservoir. And we now developing on-site rapid eDNA methods. That means that we sampling the water and filtrate on-site and eDNA extracted on-site, and we measured the DNA concentration on-site using this mobile qPCR. This mobile qPCR is very compact and quick to measure the DNA concentration. We tested this method in the river. We filtrated 250 milliliters of water, and then eDNA extraction on site. We took about 10 minutes, and then we measured this DNA using mobile qPCR. We took about 15 minutes. In total, we just took the 25 minutes to detect fish eDNA on site. So this is the result. We measured the silver carp DNA in this river and tributary and lakes. And this table showing that the eDNA detection compared with the national observation surveys. So upper means that mobile PCR on-site methods, and lower means the benchtop laboratory method using benchtop qPCR platform. We found the eDNA detection by on-site qPCR methods was similar to the laboratory method using benchtop qPCR platform. And this figure is showing the CT values of benchtop qPCR and Y is the mobile PCR. We found on-site qPCR method reported that CT values is which highly compared with correlated with the laboratory qPCR method, like this figure. So I want to introduce a sedimentary DNA for time machine biology. Sediment core analysis has been used for the time machine biology for understanding long-term baselines in community. And sedimentary DNA, SED DNA, may remain in sediment core like this picture on the water bottom. So we first tested the existence of fish DNA in the sequence of core and the relationship between the fish sedimentary DNA concentration and fish abundance. So using the sedimentary DNA in the core, we can look at recent and also 300 years ago. So this is the result. X is that years from 17th century to recent, and Y is the DNA concentration of each marine fish species. And we found the sedimentary DNA can track long-term dynamics of marine fish DNA abundance for the last 300 years. So we detected some peaks in the 100 years ago. And this graph showing that X is the landing records of these fish in Japan, and Y is the eDNA concentration of each species. So we found that sedimentary DNA concentration was, or in most cases, positively correlated with the landing records of these fish species. So that means that sedimentary DNA can track fish abundance for couple hundred years ago. So not only for population analysis, we can apply eDNA analysis for the community using the meta-barcoding. So I want to shortly introduce our meta-barcoding studies. So firstly, Dr. Mia developed the MyFish 12S primers for fish meta-barcoding. And these methods already used across the world in this, like this map. So, and we now applying the MyFish meta-barcoding methods for various aquatic habitats. This is one example, MyFish meta-barcoding for the coral fish community. We found that MyFish meta-barcoding detected coral fish more than the 300 species from just only 11-liter water sampling. And this graph showing that number of sampling sites and fish species richness, we found that MyFish meta-barcoding outperformed the capture-based surveys, like this figure. And we also tested the species detection rate by steps of the MyFish meta-barcoding. Here we evaluated 2 steps. One is the filtration steps, including the eDNA extraction, and second is the first PCR steps, like this figure. We estimated the species detection rate of these 2 steps by using the multi-species occupancy modeling. And this is the result. So species detection rate by MyFish meta-barcoding was dependent on the first PCR replicates rather than the filter replicates. This figure showing X is the number of first PCR replicates and Y is the species detection rate. And each figure showing the different number of filter replicates from 2 to 8 filter replicates. So we found that increase the number of first PCR replicates, the species detection rate increased rapidly. But among the filter replicates, the fish detection rate was not remarkably different. And very recently, we published this paper, quantitative eDNA metabarcoding by QSEC, quantitative sequence by using the Illumina MyFish sequencer. So I want to shortly introduce this method. Firstly, attach the primers for single primer extension before the first PCR. This primer, including the random sequence tags. And then this DNA with excess primers for the metabarcoding, and we analyze the number of target DNAs based on the Poisson distribution from this random sequence tag variance. And we estimated eDNA concentration of fish community in mesocosm by QSEC successfully. Finally, I want to summarize the eDNA method standardization. So eDNA Society published the English illustrated manuals for eDNA research. And also we published the commentary papers here in Environmental DNA Journal. This manual including the selection of sampling sites and water sampling and filtration using the filter cartridge and also glass fiber filters and extraction DNA and DNA analysis using the qPCR and also MyFish metabarcoding. And also recently many eDNA protocols, for example, these 3 protocols were published. However, I think the globally standardized eDNA protocols should be recommended. So I think the DNAQUA provides us good discussion place. I think especially this working group 3 session would be nice to standardize eDNA protocols. So this is take-home message of my talk. eDNA can be useful too with providing species distribution and abundance biomass by using the qPCR, ddPCR, on-site qPCR method, and also metabarcoding. This is final slide. Finally, I want to introduce you the future remarks of eDNA study. First, as I mentioned, finally, eDNA methods should be standardized globally. And the second is that genotype analysis from eDNA. So for this topic, several papers already published, and they detected and estimated genotype variance or genotype differences of species from eDNA sampling. And third is that various DNA molecular analysis, for example, the sedimentary DNA analysis and eRNA, environmental RNA analysis, and E-protein, environmental protein analysis. So some papers already published, but these other molecular methods should be developed. in future. So we now using that mostly eDNA, but in future we can use the eRNA, eProtein, and also sedimentary DNA, RNA, something like that. Thank you for your attention. Thank you.